All right, let's jump right in. Today, we're talking about one of the biggest scientific breakthroughs of our lifetime. It's a revolutionary AI called AlphaFold, and it has completely rewritten our understanding of biology.
So, let me just start with this question. What if a single computer program could solve a biological mystery that has stumped the smartest scientists for half a century? Well, that's not science fiction anymore. It's exactly what we're going to unpack today.
So to really get why this is such a huge deal, we first have to understand the problem itself. This is a challenge that scientists have been banging their heads against for 50 years.
Okay, so it all comes down to these things called proteins. The best way to think of them is as life's tiny little machines. I mean, they do pretty much everything inside our bodies, from carrying oxygen in our blood to fighting off viruses. And their function, what they do, is all determined by their very specific 3D shape.
And that brings us to the big one, the protein folding problem. See, scientists know the sequence of amino acids, the building blocks that make up a protein. You could say that's the easy part. The incredibly difficult part, the puzzle, has always been predicting how that long string of code will fold itself into a complex, working 3D machine.
And the scale of this problem, it's just staggering. Thanks to all our gene sequencing, we now have the linear codes for billions of proteins. But the number of actual 3D structures we've managed to figure out in a lab, it's only around 100,000. That is a massive, massive gap in our knowledge.
And look, this isn't just some abstract academic puzzle. This gap has been a huge bottleneck. It's held back our ability to understand diseases and to design new medicines. For decades, just figuring out one single protein structure could take years and years of lab work.
But then, everything changed, and it changed because of AlphaFold, a completely game-changing AI system that came out of the labs at DeepMind.
So what is this thing? Well, at its heart, AlphaFold is a totally new kind of neural network. What makes it so different is that it was built from the ground up to understand the actual physics and biology of how proteins fold. It essentially taught the AI to think like a physicist and a biologist at the same time.
Of course, making big claims is one thing, but proving them is something else entirely. AlphaFold had to go up against the ultimate challenge to prove it was the real deal.
And that challenge is called CASP. Think of it as the Olympics for protein prediction. Every 2 years, research groups from all over the world are given a set of protein sequences and have to predict their 3D shapes. And here's the key. It's a blind test. The real structures have been solved in a lab, but they're kept secret until after the competition.
And at CASP 14, AlphaFold didn't just win, it completely blew the competition out of the water. Just look at these results. Its median accuracy was an unbelievable 0.96 angstroms. The next best competitor, all the way down at 2.8. This wasn't just a small step forward, it was a giant leap.
The official paper put it pretty bluntly. AlphaFold was, quote, vastly more accurate than competing methods. The entire scientific community was just floored. That 50-year-old problem had basically just been solved.
To give you a sense of how insanely accurate that is, a single carbon atom is about 1.4 angstroms wide. So AlphaFold is predicting these incredibly complex structures with an error margin that is smaller than the width of an atom. We're talking about atomic-level precision here. For something like drug discovery, it's like we've gone from looking at a blurry photo of a lock to having a perfect, high-res blueprint. Now we can design the key.
So the obvious question is, how on earth does it do it? How does this AI actually work? Let's take a quick look under the hood. It's basically a 4-stage process. First, it takes the protein's amino acid sequence and digs through giant databases looking for evolutionary relatives to find clues. Next, it uses this really cool part called an Evoformer to figure out which parts of the protein are likely to be near each other. Then, a structure module takes that hypothesis and builds an initial 3D model. And here's the genius part. It then takes that model and feeds it back into the beginning of the process, refining it over and over to get more and more accurate.
The most remarkable thing is that this is a true end-to-end system. You put the genetic sequence in one end, and out the other comes the precise 3D coordinates for every single atom in that protein. It's a direct line from digital code to a physical structure.
So what does this all mean? What does this breakthrough actually unlock for us? Well, it is no exaggeration to say that this has kicked off a brand new era in biology.
I mean, the potential applications here are just mind-blowing. We can now speed up the design of new drugs like never before. We can get a much deeper understanding of diseases like Alzheimer's or Parkinson's, which are all about misfolded proteins. We could even start designing totally new enzymes to do things like break down plastic waste or pull carbon out of the atmosphere. It's incredible. We can finally keep up with the explosion of data we're getting from sequencing genomes.
The creators of AlphaFold said it best themselves in their paper. They said they hope it will become one of the essential tools of modern biology. And you know what? That's exactly what's already happening.
And the speed of this thing is just breathtaking. I want you to really let this sink in. A process that used to take a graduate student months or even years of careful, painstaking lab work can now be done by AlphaFold on a computer in a matter of minutes or hours. The leap in efficiency is almost impossible to wrap your head around.
And that brings us to the final thought I want to leave you with. For the very first time, we can accurately and quickly predict the shape of life's most fundamental little machines. The question isn't if we can know the shape of proteins anymore. The question now is, what are we going to build with this new power? The possibilities are really just beginning to unfold.